Good. You can have Dr. Mehra and Dr. Kapur come on up. We're doing something a little bit different this session, and we're going to try to replicate it in the years to come, hopefully, where we're picking out a separate section of science that's not normally highlighted at our meeting. And so this year, wiser people selected pathology. So I'll let you all take it over.
Hello, everyone. Yeah, thank you so much for drawing the curtain. So we have a lot of pathology images, so we just want to make sure they're visible easily on the screen. Welcome to all of you to this State of the Science Pathology session. I think, like Brian said, I believe a dedicated session is being done for the first time, and we are thankful to the organizers for giving us a time slot for this. I'm Rohit Mehra. I'm a geopathologist at University of Michigan Hospital. I'll be moderating this session with Dr. Payal Kapur from UT Southwestern Medical Center. So in this session, we are just hoping to share some translational pathology and surgical pathology elements from a renal cell carcinoma perspective. And this session will also be different in the way that we'll be taking questions actually at the end of all the presentations. So please note down your questions. Our first speaker is Dr. Samson from Memorial Sloan Kettering Cancer Center. Samson is a renowned urologic pathologist. He is actively engaged with the MSKCC Geopathology Group in studies defining the spectrum of tumors seen under the RCC subtypes. Samson will be presenting on the taxonomic evolution of kidney cancer with emphasis on molecularly defined RCC. Over to you, Samson.
Thank you so much, Rohit and Payal, for hosting the session, and thank you, everyone, for the invitation and the chance to address you. I want to begin also by thanking the Geopathology Group at Memorial Sloan Kettering, and specifically Dr. Xingbei Chen and Victor Reuter. Our discussions over the past 15 to 20 years have informed much of my presentation today. So this is what we're here to talk about. 2022 WHO kidney tumor classification. On the left, you can see all of the usual tumor types: clear cells and papillaries and chromophobes and some other renal tumors you may have heard of. But a lot of excitement generated around that middle column, the so-called molecularly defined renal carcinomas. And what I hope to do with you in the next couple of moments is to focus on a range of the spectrum of renal cell carcinomas while emphasizing three key messages. Number one, that while molecularly defined RCC is a new WHO category for the first time, kidney cancer classification, as you all know, but as we'll show very discreetly, has been anchored in molecular findings for greater than 30 years and formally for just about 30 years. Secondly, that morphology molecular correlation, after all, we are pathologists up here, has led to identification of new or more precisely defined entities. And also, and this has emerged much more over the past 15 years, that germline and somatic alterations in specific genes give rise to kidney cancers with morphologic overlap. So let's go here, the Heidelberg Classification of Renal Tumors, a 1997 publication based on a conference called The Impact of Molecular Genetics on Classification of Renal Cell Tumors. This is exactly what we're talking about. Our three major kidney cancer tumor types that everybody knows: clear cell RCCs, papillary RCCs, these two largely insights driven in the 80s and 90s from familial kindreds with von Hippel-Lindau syndrome and hereditary papillary renal cell carcinoma syndrome, but also known in their sporadic variants to have alterations in chromosome 3p, as well as trisomy of chromosome 717 and loss of chromosome Y, and chromophobe renal cell carcinoma from the work of Tunis and Storkel in the late 80s and 90s, then known over the 10 years after that, having loss of heterozygosity of multiple chromosomes. This classification also emphasized that sarcomatoid, I'll call it differentiation, was not a specific subtype, but rather could be present in all these major RCC subtypes, and it gave us the term RCC unclassified, which has been mutated into RCC NOS in the recent classification, for tumors that had either no recognized pattern or features intermediate between recognized patterns. Let's see some examples that came after the Heidelberg classification. So here we have a tumor that is extensively cystic. Those are the spaces. Those spaces are lined by clear cells, but there's no clear cell mass here. These were known at the time as multilocular cystic renal cell carcinoma, but over time and with greater cohorts, we know that these are indolent, and so therefore they are now known as multilocular cystic renal neoplasm of low malignant potential. They too have alterations of chromosome 3p, just like the VHL in sporadic patients. Another tumor, the mucinous tubular and spindle cell carcinoma, is originally classified as RCC unclassified because some people thought it was a low-grade sarcomatoid because there are spindle cells. And when they were crunched together, especially in the tubular portions of this, mimicked the inset, which is actually the solid variant of papillary renal cell carcinoma. But we now know has very different chromosomal pattern, which can be shown by oncoscan, SNP arrays, than papillary renal cell carcinoma. These are classic, the characteristic chromosomal losses, and occasionally we need this testing in clinical practice. And then in the early 2000s, the discovery in tumors that were papillary but were lined by clear cells and often or sometimes showed also voluminous pink cytoplasm, the XP11 translocation, RCCs, TFE3 rearranged with increasingly multiple fusion partners, which are seemingly never ending. And all of these got included a couple of years later in the 2004 WHO kidney tumor classification. In 2006, my colleague Satish Tikku published this paper about the spectrum of RCCs in end-stage renal disease, and specifically in acquired cystic kidney disease. And he showed us that in addition to clear cells, papillaries, chromophobes, and all the rest of tumors, we could see two unique tumor types. One, I think you know, is called acquired cystic disease-associated RCC, which is associated with this, might I say, cribriform, because I do prostate also, pattern, and calcium oxalate crystals. However, it also gave us this tumor, which were seen initially in end-stage kidneys in his description. These were cystic and papillary tumors lined by clear cells with a distinctive linear array. Some people have described the cells as showing a piano key-like pattern. And he called these clear cell papillary RCC of the end-stage kidneys with a characteristic basolateral or cup-like pattern of carbonic anhydrase 9 staining. Later, it was shown that these don't just happen in end-stage kidneys, but we'll call them sporadically as well. And just like the multilocular cystic neoplasm of low malignant potential, information over time has told us that these two are indolent tumors with no 3p loss, VHL mutations, or trisomy of 717, so neither like clear cells nor papillaries, and therefore in the most recent WHO, clear cell papillary renal cell tumor. Most of the other work in the past 10 years has been in this area. I'll call it renal cell carcinoma with complex architecture or high-grade cytology. We know that there are these complex papillary proliferations, very different than the typical papillary renal cell carcinomas with MET alterations. They're heaped up. You can't see it on the left-hand slide, but in the middle section with prominent nuclei. But some of these are also cribriform or sieve-like. Some of them are solid, like in the center picture. And a couple of them, or a subset of them, have these prominent nuclei with prominent nucleoli with these perinucleolar halos, which this is a perfect example, but occasionally can be much more focal. These were first noticed in the subset of cases in the context of hereditary leiomyomatosis and renal cell carcinoma syndrome, and were called HLRCC-associated RCC in the 2016 WHO, germline mutation of the FH gene. We now recognize that almost all of these have FH loss by immunohistochemistry, and there are occasional somatic cases that have been described, or at least where we don't know a history of cutaneous or uterine leiomyomas, and so therefore the preferred term now is FH deficient RCC. Also in this category were solid tubular pattern or large nested pattern carcinomas, but that had high-grade cytology. And so further investigations now created a family, not just TFE3 rearranged or XP11 tumors, but TFEB rearranged RCC, and then more recently, aggressive variant TFEB amplified RCC. And so in the most recent WHO, we have now molecularly defined entities as listed there on the bottom left. Initially, two cases described in 2006 Vinculin ALK rearrangement in patients with sickle cell trait gave us ALK rearranged RCC. You can see the complexity of the pattern in the H&E there, even if you are not a pathologist. Again, that sieve-like or large microcystic space pattern. We now know that ALK rearranged RCC can have multiple partners, and this is important because it can be targeted by ALK inhibitors. And finally, in tumors that very often morphologically overlap with that, we've known about renal medullary carcinomas for many, many years. But now we know that these patterns, which have nests and cribriform and diffuse growth, and are associated with neutrophilic abscesses on the bottom there, and sickle cells, because some of them occur in sickle cell trait or other hemoglobinopathies, have loss of INI1 or SMARCB1 and are now referred to as SMARCB1 deficient renal medullary carcinoma. Oncocytomas are our best known benign tumor in the kidney, and we also know that there are other eosinophilic or oncocytic pink cell tumors. The most common of those in the middle there, the chromophobe, and I'm showing you an eosinophilic variant of chromophobe, which doesn't always have all the chromosomal losses of its partner. But to the left are a series of tumors that looked a lot more solid, though having pink cytoplasm to the pathologist. And eventually these were worked out as being almost uniformly present in germline SDH mutated RCCs in the context of SDH deficiency and the hereditary paraganglioma pheochromocytoma syndrome. On the flip side of that, we can see oncocytomas, chromophobes, eosinophilic variant of chromophobes, and eosinophilic cysts in Birt-Hogg-Dubé syndrome, and specifically in a condition called renal oncocytosis, where you can see up to hundreds of small lesions in patients. Specifically in Birt-Hogg-Dubé syndrome, with the gene folliculin, we've also seen described mixed oncocytoma chromophobe renal cell carcinoma feature tumors, and these hybrid oncocytic tumors, which seem to have clear and pink cells. The spectrum of findings on the right here has also been described in tuberous sclerosis-associated renal cell carcinoma, which has a wide variety of renal manifestations, the most common of which is angiomyolipoma. In the renal tumors, though, in addition to ones on the previous slide, descriptions in the 2010s grouped TSC-associated renal cell carcinoma into three groups. The first they called hybrid oncocytic or chromophobe-like tumor, very descriptive. And they also mentioned this thing called the granular eosinophilic macrocystic tumor. Now, it's very interesting because we have recent tumors included in the WHO called eosinophilic vacuolated tumor, low-grade oncocytic tumor, and eosinophilic solid and cystic renal cell carcinoma that my colleagues on stage here, Drs. Mehra, Kapur, Hirsch, and my colleague Yingbei Chen, have now shown to be sporadic renal tumors with somatic TSC-mTOR mutations. So similar, same genes, similar morphology between hereditary and somatic. And I'll end with this slide, which is the last tumor that was included in the TSC-associated renal cell carcinomas in the 2010 publications, and that is renal cell carcinoma with fibromyoma stroma. Now, this has been recognized for a long time and was included as a provisional entity there on the left in the WHO 2016 when we thought this was a descriptive diagnosis. A lot of older terminology. These are tumors that have clear cells, can show papillary or undulating there on the right morphology, but are characterized by large nodules with broad fibromuscular bands and embedded in this thick fibromuscular stroma. We now know that these tumors can occur in TSC-mTOR altered RCC, either in the germline or in the somatic setting, and are also associated with ELOC, or what used to be known as TCEB1 mutated RCC. So in summary, kidney cancer displays tremendous morphologic and molecular heterogeneity. Classic morphologic manifestations can be clues to the pathologist to appropriate workup, so we don't do the kitchen sink, as it were, on every tumor. Review of subsets and larger cohorts with profiling, there's going to be an increased likelihood of identifying these, but I'll add, as clinicians, we know that the findings should be placed in a clinical context. We should be able eventually to divide these into indolent tumors, those with intermediate risk, and those that are more aggressive. Thank you very much.
Dr. Samson. Our next speaker is Dr. Michelle Hirsch. Dr. Hirsch is Chief of GU Pathology at Brigham's and Women's Hospital. She will be talking about surgical pathology toolbox and technological advances to solve kidney cancer problems.
Thanks very much, and thank you for the invitation to speak today and being part of this meeting. I've been with KCRS before, and it's always one of my favorite meetings to attend, so I'm happy to be here. As mentioned, I'm going to be talking, Samson went through sort of how we classify tumors. I'm going to be telling you how we did that. What are the tools and the ancillary tools that we can use to get to these diagnoses for you and your patients? So I think it's important to understand why we do this. We can talk about splitting and lumping tumors into big categories, and what we would like to say, I think as pathologists, and I think many of you would agree for your patients and how you care for your patients, that getting to a specific renal cell cancer subtype has prognostic, therapeutic information that is useful for you when you're deciding how to treat your patients. But I think it's important, what we're trying to do is give you the background, so I'm sort of breaking it down into what is a diagnostic tool that we might use, and it's a process of evaluation which is used to see similarities and or differences in a tissue on cells and or cell components. And so what are some of the tools that we can use to do this, to see these different parts of the cells or parts of the tissue? And we look at them in different ways. We can look at structural entities, structural portions of the cell or tissue, and mostly we use H&E stain, and that is the most common tool that we use every single day to look at these tissue types. Traditionally, and many, many years ago, we would use electron microscopy to look at a much deeper level of the tissue, and we have special stains which are used occasionally. Protein expression can help us a lot. We can look at different things within the cell, transcription factors, proteins, et cetera, and we do this by immunohistochemistry, and we'll talk a bit more about that. We can also look at genetic and molecular alterations using a whole host of more contemporary types of studies. This would include SNP microarray, conventional karyotype or cytogenetics. We can do next-gen sequencing, et cetera. And again, we'll talk about these very briefly in a little more detail. What can we see with all these tools? Well, just those things that we're looking for: structural components, proteins, cell adhesion molecules, DNA, RNA, et cetera. And how are these tools helpful? Well, we use them at the research setting, of course. We can just take a whole bunch of kidney tumors and apply these diagnostic tools, and that helps us separate them into first research approach to looking at separating or splitting these tumors into different types, and then hopefully that translates into a clinical use of these tools, which helps you, again, improve taking care of your patients. Of course, there are also diagnostic and tools or special ancillary studies that we can use for both prognostic and therapeutic reasons. So again, as mentioned, Samson already went through the WHO classification really nicely in just a few minutes, but these are, again, this is an example on the left-hand side of what I've just explained, all these different types of tools, which have ultimately led to the current 2022 WHO classification of kidney tumors. And again, I mentioned already that H&E stain is our gold standard. We are morphologists, and when you have a kidney tumor with perfect morphology, you can actually make the diagnosis on H&E alone. It's the quickest, cheapest, and easiest stain to use, and again, in many cases, or a subset of cases, we can make the diagnosis. These are 8 examples, perfect examples that could be diagnosed by H&E alone. The problem is there's a lot of morphologic overlap in other kidney tumors or in subsets of each of these specific subtypes that Samson has talked about. And here's 2 examples of 2 images from 2 different kidney tumors. The left is a clear cell renal cell carcinoma, and on the right is a translocation TFE3 XP11 renal cell carcinoma, which essentially look identical. And it's up to us to be able to know when we need ancillary tools to use and separate these by able to make the correct diagnosis for you. So that on top of the H&E, we use that to form our differential diagnosis. Immunohistochemistry is the most frequently used tool, and basically this is using an antibody to attach to a protein, and we can then look at it with light or fluorescent microscopy to look at various features and different staining patterns to help us make a diagnosis. It's simple, it's cheap, it's quick, and it really gives us a lot of information. So in this particular case, on the left, it had CA9 expression, which confirms clear cell renal cell, and on the right, TFE3 nuclear expression, which confirms translocation renal cell carcinoma. And we can take all the information we put together and develop algorithms or flow charts like this, and this is just to sort of show you an example. By using positive and negative staining patterns, we can get to specific diagnoses. And with that information, you can then subclassify these kidney tumors, and here's just the 4 most common types of kidney tumors that we see, showing you gross, histologic, immunophenotypic differences between these types that we learn to know. And on top of it, on the far right-hand side, you can see that they have genetic differences as well. Traditionally, that was found, we could use karyotype or G-band karyotype cytogenetics to distinguish these tumors. That isn't used as frequently anymore because we can use other tools in its place. Cytogenetics requires fresh tissue. We can now use paraffin-embedded tissue for a lot of these studies. In situ hybridization is, in a sense, similar to immunohistochemistry. It's just looking at DNA and RNA hybridization instead of an antigen and an antibody. But ultimately, there's a probe or a label on this hybridization technique. The details of the technique are not so important, but we can look at with a chromogenic or a fluorescent tag and be able to look at these cells. And the most commonly that's used right now in breast cancer, of course, looking at HER2, you can use immunohistochemistry to look for HER2 expression, and in situ hybridization is looked for HER2 amplification. But in kidney tumors, we can use that using fluorescent microscopy to look at 3p loss. The loss of a green dot there is indicative of 3p loss, or extra chromosomes 7 and 17 in a papillary renal cell carcinoma. We can also use this to look for translocation renal cell carcinomas in break-apart assays. So there are some more contemporary studies, and actually many of these have been done, and Rohit has been the one to publish these, where we look at RNA in situ hybridization to help us when we have difficulties in a subset of cases. This is a fluorescent break apart in the upper little small box showing you the break apart for a TFE3 translocation renal cell carcinoma, and he has shown that RNA in situ hybridization has higher specificity in some of these cases with a TRIM63 RNA in situ hybridization. The challenging part with these studies, although they're really great, is that not many laboratories or not many institutions have these probes, so you have to be someone to take the time to invest in these, and I just think that they're potentially a way for the future, but most laboratories don't have these in situ hybridization probes. Great, though, for progress. So the other thing we look at is single nucleotide polymorphism. We can look at copy number analysis. I showed you earlier, karyotypes can be used to look at the different genetic alterations in specific types of kidney tumors. That was fresh tissue. In paraffin-embedded tissue, we can do SNP microarray to look at chromosomal changes to help us with a diagnosis. It's a little bit of a longer turnaround, 1 to 2 weeks over immunohistochemistry, but if we can't get to a diagnosis with immunohistochemistry, we find this quite useful. And this is a case here of a 60-year-old man. It's a tubular architecture, which would make you think about mucinous tubular and spindle cell carcinoma, but those usually occur in young females. So it's sort of the wrong demographic. So if we can't get to the diagnosis, we can do SNP microarray. This particular case had the multiple monosomies, or loss of multiple chromosomes, confirming the diagnosis of mucinous tubular and spindle cell carcinoma, and excluded the diagnosis of papillary renal cell carcinoma. The other tool that you're probably all familiar with is next generation sequencing, and this is a high throughput assay that looks at, usually in most institutions, targeted genes, and is really looking much more for mutational analysis, but it also gives you the copy number variation that I was talking about with cytogenetics or SNP microarray. It detects single nucleotide variants, insertions, deletions, copy number alterations, and structural variants. And this is just an example of one plot of 30 cases where we were looking at. On the left-hand columns, you can see the mutational analysis. On the right-hand, plotted out are the copy number variations. Now, we don't use this routinely diagnostically, and part of that is because not every laboratory, again, has access. Sometimes you have to send these out. They do take time. They're costly. And at the end of the day, the mutational analysis isn't always giving us the final diagnosis. So I think that this is used more frequently in tumors where we get stuck and have an unclassified renal cell carcinoma, or if you're really looking for a specific mutation for therapeutic reasons. So it's not standard day-to-day day-to-day diagnostic ancillary test, but certainly is performed not infrequently. And this, just to end up here, is to show you an example of a case. This was a large kidney mass with multiple metastatic sites in a young woman, and the H&E is not specific for a diagnosis. A battery of immunostains, fluorescent in situ hybridization was done, basically excluding on the right-hand side a whole differential of diagnoses, and we basically ended up with unclassified RCC, and at the end we sent this for next-gen sequencing, and it ended up with having a TSC2 mutation, but did not have a specific type of diagnosis, so may have been helpful in that regard. So my last slide is to say, should we do molecular workup for all diagnoses of kidney cancers? Well, on one hand, you can say, why not? It's done in other subspecialties, but it is expensive. Again, not all laboratories have it, and it may require a send-out. The bottom line, I would argue it's not necessarily needed. Of course, that last case I just showed you, we did need it. But in most of the time, we can do what we now call next-generation immunohistochemistry. And we have a whole lot of biomarkers that really get us to the end diagnosis. The simplest and most common is the use of CA9 to confirm clear cell renal cell carcinoma, although not entirely specific. And here's a whole other list of antibodies that we can simply do very quick, very cheap immunohistochemistry on to give us a lot of information for you to treat your patients, including the loss of SDH or the loss of FH to suggest that a patient might have a familial cancer, or Merlin loss to say the patient has an NF2 mutation or a SMARCB1 deficiency. So this whole group of antibodies has been really helpful and helped us to classify these tumors for you as best as possible. And with that, I thank you for your attention.
Thank you, Michelle. Our next speaker is Dr. Payal Kapur. Payal will be talking about prognostic and predictive biomarkers in kidney cancer.
Thank you, Rohit. Delighted to be here. So numerous prognostic and predictive biomarkers have been investigated in RCC over the last couple of decades. These include individual or combination of clinical, tissue-based, sequencing-based, liquid biopsy-based assays. However, only a few are used in the clinic. This is an active topic of research, especially in the context of immune checkpoint inhibition prediction. We and others in this audience have shown that BAP1 loss is a very robust prognostic biomarker. However, especially in the context of small renal masses. However, today what I would like to do is focus on two short projects illustrating the power of one of the most ubiquitous and inexpensive biomarker, morphology. Clear cell RCC exemplifies intratumoral heterogeneity, and as you can see here, there's quite a bit of heterogeneity. Today what we do is we go to the highest grade area and we ignore the background morphologies. To investigate what the role of this intratumoral heterogeneity was, we investigated 550 consecutive clear cell RCCs in order to deconvolute the complex phenotype of clear cell. We interrogated these tumors along 3 axes. Architecture, cytology, and microenvironment. So we evaluated these 550 tumors for these 33 parameters. Even though there was extensive intratumoral heterogeneity, we found these 9 architectural patterns to be frequently occurring in these tumors. The frequencies are shown in this figure. We found that 86% of the tumors showed 2 or more patterns. The most common pattern was the small nest pattern shown on the top, which is the pattern we use for making the diagnosis of clear cell RCC. And the 4 patterns shown on the right in red were irrespective of the amount of that pattern or the other patterns present in the tumor, were associated with aggressive prognostic variables, including grade, stage, and development of metastasis, and with outcome. And some of these variables were significant even in multivariate analysis. We found that these aggressive patterns had progressively spaced out vasculature compared to the indolent patterns. They were often next to these indolent patterns, were present in smaller numbers and in larger tumors, suggesting that there was an evolutionary process here. Considering that these 550 clear cell RCCs were in different stages of evolution based on the architectural composition, we plotted a Tisney plot. So here what you're seeing is each tumor is represented by a dot, and the color is based on the most dominant pattern. So basically tumors with similar composition are clustering together. In order to get the direction, we overlaid them with the grade and size, which is unidirection. And indeed, the patterns which had the most spaced out vasculature were the more evolved patterns. Considering that tumor thrombus forms at a later stage of evolution, we evaluated the patterns in the thrombus and the paired primary tumors. And indeed, we found that there was a preferential selection of the more advanced phenotypes in the tumor thrombus. We were able to validate this in a separate cohort of tumor thrombus clear cell RCCs. And in this cohort, we found that the grade of the thrombus and not of the primary was the strongest predictor of metastatic free survival in multivariate analysis. This data has now been confirmed by a multi-institutional cohort of over 600 PT3 clear cell RCCs, and also several other groups using our vascular-based architectural classification have shown that it it predicts outcome beyond the current models such as Science Score and UCLA. I'm going to shift to my second study, which is based on digital pathology modeling. This was funded by the DoD and was co-led by Satvik Rajaram, a computational biologist. And most of you in the audience are familiar with the data from the iMotion 150 and 151, which through gene expression showed that clear cell RCC could broadly be classified into two clades of tumors. The angiogenesis-rich tumors, which had high angio score and CD31 IHC expression, and the proliferative clade, which had a subgroup of cluster 4 with high T effector gene expression and increased CD8 IHC. However, RNA-based assays are difficult to implement in the clinic. Here, for example, you can see 4 cases. Depending on the area that was sampled, the angio score can be widely different. So we asked that given that the vasculature and CD8 is so easy to recognize on an H&E slide, can we build two deep learning models that could predict the angio score and the T effector score from the H&E slide itself? And based on these scores, the appropriate therapy combination could be used. So for the angio score model, we built this giving it data where we had both the RNA or the CD31 IHC. And in order to make it interpretable, we asked that it make a vascular mask based, which was the basis of its angio prediction. We applied that to a cohort where we had multi-regional sequencing. For example, here you can see that in this block, 3 areas were punched. 2 are from the high angio score area, 1 from the low in orange there. And indeed, the model was able to predict with quite high accuracy. Once we were satisfied with these results, we applied it on multiple different cohorts. And as you can see here, it was able to predict the angio score from just a single glass slide with very high concordance, the highest in the iMotion 150, where the image and the tissue for the RNA matched with a correlation of 0.8. And this vastly outperformed the CD31 IHC score, which was also used in iMotion. So we wanted to look for what were areas of discrepancy. And as you can see here, the tumors which had both high angio score and DL prediction had very high vasculature, while the ones at on the left side on the top, the vasculature is low and the predictions and angio score both are low. But for the tumor shown in green, where the angio score RNA based was very high, but our model predicted very low, when we went and looked at the H&E, indeed the tumor is very low in vasculature, but there were a lot of vessels around the tumor, which may have influenced the RNA-based angio score. Having this robust assay, we went and applied it to multiple larger cohorts where we didn't have RNA, and indeed we found that the angio score inversely correlates with grade, stage, sarcomatoid status, and BAP1 mutation loss. But more importantly, the model can predict frontline anti-angiogenic therapy response that rivals the performance of RNA-based angio score, as shown here in the iMotion 150 clinical trial. We are now finalizing the second model, which is able to predict T effector score. Here you can see the H&E slide is marked by the model. The red are the lymphoid cells, the green are tumor nuclei, and the blue are vasculature. In the UT Southwestern cohort, the concordance is pretty high, and in the iMotion 150, it's able to predict immune checkpoint inhibition response. And in the interest of time, I'm going to just silently acknowledge all the people in my team. Thank you, everyone.
Thank you, Payal. Our next speaker is Dr. Sabina Signoretti. Sabina will be talking about diagnostic updates and therapeutic implications in sarcomatoid RCC, a very important topic which has already been mentioned several times today. Thank you, Sabina.
Thank you so much. I saw that picture of me. I look like a baby. Thank you. So I have, I guess, 8 minutes, not 7 minutes, to talk about this. So I'll do my best. So I'm just going to start by reminding you all that according to the WHO guidelines, sarcomatoid renal cell carcinoma is not an histological RCC type. And in fact, sarcomatoid dedifferentiation can be seen in many different types of RCC and occurs in about 5% of all the cases. It is defined histologically by the presence of these, I guess, no, sorry. Won't play with that. With the presence of atypical spindle cells that resemble a sarcoma, as you hopefully can appreciate in this microphotograph here. And what's particularly interesting is that a minimal proportion of sarcomatoid component is really not required for the diagnosis. And when this component is present, it is categorized as a grade 4 according to the WHO classification. Although tumors with pure sarcomatoid morphology certainly do exist, in a majority, in most of the cases, the sarcomatoid component is not the predominant component of the tumor and can be also relatively small. So, and this is exemplified here in this image of a large golden yellow mass of the kidney that you can appreciate. Do you know how to activate the? No. The pointer. Oh, no, it's working. Okay, so you can see this golden yellow mass of the kidney, which is characteristic of a clear cell renal cell carcinoma, with the presence of a relatively small nodule that is highlighted here in C, that has more whitish grayish appearance. That is an area of sarcomatoid growth. And given the heterogeneity in the distribution of the sarcomatoid component, percutaneous biopsy is really not accurate for making the diagnosis of sarcomatoid RCC, and this diagnosis still really relies on the pathological analysis of resected tumors. Historically, sarcomatoid renal cell carcinoma has been associated with a very poor prognosis. Unfortunately, most of the patients really already have metastasis at presentation, and the response to chemotherapy and targeted therapies is really usually very poor. And in the past, the survival has been only about a year or less. And also there is substantial evidence that the higher the amount of the sarcomatoid component, the worse the prognosis, at least in the past. Fortunately, the situation has changed dramatically in the past several years with the utilization of immune checkpoint inhibitors. And multiple clinical trials of immune checkpoint-based therapies in metastatic clear cell renal cell carcinoma that include the Checkmate 214 trial have demonstrated that in sarcomatoid patients that treated with immune checkpoint inhibitors have significantly prolonged survival compared to patients treated with VEGF-targeted therapies. And real-world data analysis that has been performed recently by Tony Chueri's group seem to suggest that the survival benefit with immune checkpoint inhibitors versus targeted therapies can also be seen in patients with non-clear cell sarcomatoid RCC. But this, I think, needs to be further evaluated in the context of clinical trials. Now, correlative analysis of clinical trials have demonstrated that sarcomatoid RCC displays an inflamed phenotype and providing some explanation as to why these tumors actually do respond relatively well to immune checkpoint inhibitors. And for instance, in the IMMotion 151 trial, the sarcomatoid tumor have been shown to have enrichment in various transcriptional pathways that are involved in immune response. As you can see here, there was also significantly higher levels of TF factor and myeloid inflammation gene signatures, and similar results were confirmed then independently in other clinical trial cohorts, including a study from our group. There is also substantial evidence that sarcomatoid RCC has increased levels of expression of PD-L1 compared to non-sarcomatoid RCC. And in a relatively small subset of cases, about 6%, this is at least what's been reported, the tumor cell PD-L1 expression is driven by amplification at the 9p24 locus that spans the PD-L1 gene. And in addition, I would like to highlight that some more recent spatial transcriptomic profiling efforts that are from the University of Michigan are starting to reveal the crosstalk between the immune cells and actually immune cells, particularly myeloid cells, that contributes most likely to the immune phenotype of this tumor. So much more to come, hopefully very soon from this very interesting area of investigation. In terms of genomics, multiple investigators have tried to identify genetic alterations that drive sarcomatoid differentiation in clear cell renal cell carcinoma. And this has been done by sequencing DNA that was extracted from tumors from areas of epithelial morphology or sarcomatoid morphology from the same tumors. So these studies have shown that the sarcomatoid component was somewhat enriched in p53 mutations. However, the p53 mutations were present only in a subset of these sarcomatoid cases that were analyzed. And moreover, pretty inconsistent results were obtained regarding mutations in other genes. So overall, no definite mutational drivers of sarcomatoid differentiation have been identified through these efforts. However, one of these studies that I would like to highlight that was actually co-led by Gabriel Malouf in France and Tori Ariakimi, who is here, identified mutations in NF2 and other genes in the hippoyaptase pathway in a considerable percentage of these sarcomatoid, clear cell sarcomatoid RCC cases. And in this study, the mutations were actually found in both the epithelial and the sarcomatoid component. Now, as you might know, mutations in these genes are not specific for sarcomatoid RCC. In fact, especially NF2 mutations can be found in different types of renal cell carcinoma, including aggressive types like unclassified RCC. But I think they are of particular interest because there are some functional studies that seem to suggest that NF2 deficiency in RCC cells really confers dependency on the downstream effectors of this pathway. And pharmacological inhibitors of these effectors are currently being developed, and some are actually being evaluated in early clinical trials for other type of tumors, mostly mesotheliomas, that have a high rate of mutations in NF2. But this opens the possibility for future therapeutic approaches for those patients with the renal cell carcinomas that harbor mutations in this pathway. And I guess I'm over my time, but this is my last slide. And I would like to just briefly highlight some more recent efforts that have been directed at characterizing the epigenome of sarcomatoid RCC. This is work that has been led by Sylvain Baca in our cancer center. This work that has been just published not only identified that some candidate transcription factors that are potentially driving sarcomatoid differentiation in clear cell renal cell carcinoma. But what I would like to stress is that they also, this work also was able to identify epigenomic signature of sarcomatoid differentiation in circulating DNA, open the possibility of developing a non-invasive blood-based diagnostic test for sarcomatoid tumors, which could be very beneficial for our patients. And I believe that tomorrow you're going to hear much more about these in one of the presentations. So and I think this is pretty much what I have today. And thank you for your attention.
Rohit, Mehra, and he's professor of GU pathology at University of Michigan, and he'll be talking about the role of pathologists in today's kidney cancer diagnosis and research.
Thanks, Payal. We are a little bit over time, so I think I'm just going to keep it brief, and hopefully we will still have some time for questions. So I think we have all talked about intense morphologic diversity in between different renal tumors. So these are all 6 WHO confirmed renal tumor subtypes, and under the microscope, they look all very distinct and very different. The questions we can begin to ask are, or what we have been trying to ask in this meeting and otherwise are, what are we seeing, and why are we seeing this, and can we use it to our clinical advantage? So I think, like everyone knows here, the human kidney has about 900,000 nephrons. Each kidney has about 900,000 nephrons, which forms the histologic, functional, and physiologic unit of the kidney, each nephron. Based on single-cell sequencing experiments, we know that there are about 50 different cell types within the kidney, and there are specific cells along the epithelial lining of nephron, and each cell carries a potential for neoplastic transformation. We have actually been able to do single-cell sequencing experiments, and what we have learned is that the common renal cell carcinoma subtypes, for example, the clear cell RCC, papillary RCC, originate from the proximal nephron, whereas most of the pink tumors or the oncocytic tumors of the kidney tend to originate from the distal nephron. Actually, now we are able to perform nephronal mapping. And what that means is that we are able to label different anatomical segments of a nephron with specific biomarkers, which are actually listed in this in the bottom right side. And we can label these different nephronal segments with these biomarkers. And these biomarkers have been nominated based on single-cell sequencing, other platforms. And the advantage is twofold. One thing is we can actually determine the cellular origin of tumors, and second is we can determine cancer-specific biomarkers. We can actually now indeed walk through the nephron from the proximal to the distal end, and we can actually, or we have what we have figured out, or we have been able to figure out the cell of origin for most of the renal tumor subtypes. For example, the clear cell RCC, papillary RCC, predominantly MITF RCC originate from these cells, which are the uncommon PTB cell clusters, which have been named differently in different studies. These cellular clusters reside in the proximal nephron. And then most of the oncocytic tumors arise from these distal segments of the nephron. So let us review clear cell renal cell carcinoma, commonest renal cell carcinoma subtype in adults. And we believe that this tumor actually arises from these, again, from these PTB cell clusters within the proximal convoluted tubules. With the aberrant VHL pathway and the canonical copy number changes, like everyone knows here, there's an overexpression of HIF target genes, which leads to heightened cellular effects like angiogenesis, glycolysis, apoptosis. And we can see these effects under the microscope. This is a clear cell RCC. Angiogenesis manifests as this racemose vascular network. Glycolysis, apoptosis leads to increase in glycogen as well as lipid content of these cells. And that is why we see this optical clarity under the microscope for clear cell renal cell carcinoma. Let us review MITF RCC defined by rearrangements of the TFE3 and TFE-B genes. Not all of these tumors, but the majority of these, again, arise from the proximal nephron, similar PTB cells. The current diagnostic category of MITF renal cell carcinoma includes TFE3 translocation renal cell carcinoma, TFE-B translocation renal cell carcinoma, and the more recently described TFE-B amplification renal cell carcinoma, which is very important to identify, the reason being that these are generally associated with poor outcome. So FISH is considered to be the diagnostic modality of choice, and this recent FISH-based survey we have done of more than 450 tumors submitted to us with a suspicion of MITF renal cell carcinoma, we found this poor prognosis associated TFE-B amplification cases to be at least 3 times as common as the TFE-B translocation cases. Based on RNA-seq, we have also been able to identify a marker called TRIM63, and this is the results of the TRIM63 RNA-ish, which we can see over here. So we can see this brown granular dot-like expression, and the power of this technology is that we are able to visualize RNA transcript directly under the microscope. So each brown granular dot we see here actually is the TRIM63 RNA transcript itself. So we have been using this clinical assay for 2 years now in our IHC lab, and we find it to be very helpful. It is like any other, this ISH assay is like any other IHC assay, like we bring on any other immunohistochemical antibody. It's very, very straightforward assay to bring on. It's a robust assay, so I would recommend please try it on. I'll conclude by sharing this case history of a 59-year-old gentleman who presented with a large cystic mass for which the patient underwent a cystectomy. This is the low-power view, the background renal parenchyma, and the patient had a large cystic tumor with multiple mural nodules, and this is one of those mural nodules which was present. So low-power view showed a complex tubulocystic and tubulopapillary growth pattern, and the referring pathologist had rendered a diagnosis of collecting duct carcinoma. But in the high power, we can see the features which have been mentioned before, large nuclei, perinuclear halos. And I will remind this patient did not have any previous syndromic history. So I performed FH immunohistochemistry, which showed that the neoplastic cells had loss of FH expression. So based on these features, we enrolled this patient in our next-generation sequencing, or our MyOncoSeq program, which is our in-house next-generation sequencing program, which is also driven by Dr. Alka Vaishampayan, who is in the audience. And MyOncoSeq analysis showed a germline variant in FH associated with a loss of heterozygosity in tumor. And I was able to render a final diagnosis of HLRCC-associated renal cell carcinoma for this patient. So since then, now we have found this patient's family members to have several stigmata associated with HLRCC. So I firmly believe that a pathologist is not uncommonly the first physician to encounter signs suggestive of hereditary renal cell carcinoma. I have been in this situation myself several times. So I believe the overarching goals for renal tumors, we should be, or we have been able to, or we need to be able to find a specific cell of origin, link it with its unique clinical and histopathologic features, find pertinent biomarkers, find the genetic, genomic, metabolomic features of each tumor, finding the copy number patterns is very important. Actually, we can diagnose almost every renal cell carcinoma just by copy number pattern. And then, of course, as pathologists, we want to be able to help and assist our clinical colleagues to be able to perform better and molecularly informed clinical trials. So I believe the functions of a pathologist have evolved considerably from their focused foundation function as a microscopic pathologist to now acting as a consultant and translational pathologist member of a multidisciplinary oncologist team. As pathologists, we are performing autopsies. We are involved in clinical trials, animal models, biomarker workup, R&D, translational research, developing AI models, as well as other functionalities. So I can speak for the members of my panel here that this is an exciting and a fantastic time to be a pathologist. We love working with all of you, and I hope all of you are having fun and a good time working with your pathologist as well. Thank you for your attention. Over to you, Payal.
Thank you, Rohit, for that wonderful talk. Our session is now open for questions.
Here.
Hey, just a question that I heard may have become an issue lately for pathologists, which is I think that the FDA has changed the bar for some of the tests, right? So before I think you could get away with some like clear, things that I don't understand, clear approved tests, and now there needs to be FDA approved, a different bar. And so in your daily work now, has that affected you? Let's say for your TRIM63 assay or something like that, or what's the state?
Is that working? Better. So it's a really good question, and it's come up, and we talk about it at our faculty meetings. Right now, we haven't had any definitive limitations, but we do know it's sort of potentially on the horizon. I think where it may have a bigger effect is the big molecular clinical testing and laboratories. In effect, it could affect our immunohistochemistry laboratories as well, but we don't have anything that stopped us at the moment. So right now, I know it's different at different institutions. At the Brigham, we cannot order next-gen sequencing as a diagnostic test. When I want it or think a tumor needs it, I call my colleagues and say, hey, order OncoPanel. And they do. They're great. They'll just, but I can't order it from a diagnostic. That has nothing to do with the FDA. It's just it's the setup in our institution, which is different than other institutions. But the other, FISH, SNP, all those things, I have had no problem as of today ordering any of those tests. But we're keeping an eye on what the FDA is seeing.
If I could just add something to that. No problem. Just to add, especially in tumors with complex architecture that don't, immediately hit the eye as one of the usual types, we're often left doing, as you've heard, a panel, like almost a kitchen sink worth of markers and the like up front to try to divin out and get more precision. So we would hope that wouldn't go away as we've developed all these tests.
I think that, there is some hope for the future. I think the FDA decision is not so categorical that the test would have to be FDA approved, that there'll be some regulatory elements, but I think we hope that we'll be still able to work with our homebrewed tests.
Yeah.
At least, it'd be hard. A lot of tests might be grandfathered in. It'd be hard to develop new tests and continue to keep up with the industry and the competition of, Foundation One and things like that. So right. I think it's a big issue for academic centers that invested so much into building up molecular programs.
Got it. The question that came up, or how it started, so to say, was discussion at my own institution because we were impressed with some of the KIM-1 data that were presented, and we wanted to, get it into our institution to learn more about it and test it on some of our patients. What's the status of KIM-1 in your institution, and do you see this being like a roadblock to implement it other places?
I think you need to start to speak to your the person
What's
in
that?
the right side.
Yeah.
I think that
most laboratories don't have it for a clinical diagnostic purpose. It's a research antibody. What we do with research antibodies, and we could talk about a number of them. For example, SOC17 is an antibody that I have looked at the research level. There's literature about SOC17. It's mostly for GYN, but can be used in germ cell tumors and such. And we run a validation. I just go to the person who runs our laboratory and I say there's literature on this, I've done cases, and then we can bring it on. So I just have to give him some positive and negative cases and prove to him that it works efficiently, and then if they give me the okay, I can bring it on as a diagnostic clinical biomarker. So most people don't, that I'm understanding is most people don't have KIM1 as a diagnostic biomarker, but in theory, if we wanted to bring it on, we would just have to validate it in our clinical laboratory, prove that we think it works, that it has good specificity and sensitivity, and then we could bring it on and use it diagnostically.
Thanks, Michelle. Please go ahead.
Yeah, this is a fascinating session. I really enjoyed it.
Thank you.
I think the five of you are the experts in the field, and I think it's time to really have a consensus about when you call sarcomatoid, Sabina. I see you say dedifferentiated or differentiated, so it may be semantics, but I think it's time to really, use the same term. So across, the institutions, what you mean by sarcomatoid, dedifferentiated or differentiated. The other thing that is commonly reported by, in the pathology reports at our institution, of the Emerson and many institutions, is rhabdoid features and sarcomatoid features, and it's confusing. I think it's time to also look, I think sarcomatoid is different than rhabdoid, and I think we need to look at it. We could look at it like we did with the sarcomatoid Sabina and Checkmate 214, and ask BMS that we should do that to really see if rhabdoid alone is grade 4, sarcomatoid alone is grade 4. Is the combination, when you see mixed rhabdoid and sarcomatoid, does it make a difference? Is it worse than sarcomatoid? I think we really need to get to it. So the other comment I didn't hear you talk about this much, and I think collecting duct, Bellini's tumor, is really an orphan disease. And there is a divide across the Atlantic between the US pathologists, rarely report collecting duct. At MD Anderson, I've been there 23 years, I can count a dozen cases of collecting duct. Europeans, in Italy alone, they have 50 cases. I just don't think we have a firm way of making the diagnosis of collecting duct, and because there is really no specific biomarker. It's not like RMC, or you have SMART-B1 and you have sickle cell trait. And then, of course, with TFE3, you spoke about that, TFE3, TFEB, rearranged and amplification. Can you speak about collecting duct and guide us how to really make that diagnosis? Because there are patients, there's really no good therapy for this, and we really need to really...
Yeah, perhaps for sake of time, I can take this one. I think collecting carcinoma, it still exists in the WHO. I do not think there is a consensus yet whether to totally exclude it or not. I personally have not made the diagnosis in 5 years. Number one. Second point is Foundation Medicine, like everyone knows, did a study on collecting the carcinomas, and 2/3 of those patients had FH loss. Does that tumor exist? I'm not certain. It's an adenocarcinoma NOS. I think it needs more distinct studies. Till then, I'm very personally hesitant to make that diagnosis till so far. But I think if I see a good study coming out, which I believe in, I think that might change. And I think the reason I'm not passing this question on to my colleagues is we also don't have consensus amongst ourselves. There's going to be some discrepancy. Your points are very well taken, though. They are like right spot on. Thank you. Please go ahead.
Well, very well taken about the sarcomatoid. I didn't have the time to discuss the fact that there is not real 100% consensus of what pathology still nowadays call sarcomatoid. So it's an issue. It's an issue when we try to treat this patient differently because there is no agreement on the pathological, diagnostic criteria. But
thanks, Sabina. Go ahead. Please go ahead.
Have you had a question for Dr. Kapur? Have you had the opportunity to probe for an interplay between the angio score and either a response to IO or the tumor immune microenvironment?
So have you had the opportunity
to probe between any interplay or correlation between angio score and IO response or tumor immune microenvironment.
So we've just about finishing up our T effector one, and then we were, then that's the next logical step, and also to go into real world data and see how that pans out. So absolutely, another immune checkpoint inhibitor. So that's our next step. Thank you.
Sorry if we are out of time, but all of us will stay over here for any other questions there are. Thank you so much for your attention.